This video focuses on providing an introduction to enzymes, which are proteins catalyzing chemical reactions. Enzymes are essential to bodily function. In fact, life would cease in a matter of less than a second if our enzymes failed to work. Enzymes allow the body to control the rate at which chemical reactions occur, thus governing important processes such as metabolism and a variety of other aspects of our life responsible for health. In this video, we give an introduction to enzymes, including some of the key terminology that we'll be applying in later videos. Later videos will focus on covering more details about exactly how enzymes work, what different categories we can place enzymes into, and topics related to how enzymes are inhibited by drugs and other molecules. Now let's talk a bit more about what exactly are enzymes. Enzymes are a special category of proteins that act as catalysts for chemical reactions. Catalysts, by definition, are molecules that speed up the rate of a chemical reaction without being consumed or destroyed by that reaction. In other words, enzymes are recyclable catalysts that can catalyze thousands and thousands of reactions within the period of a second or less in many cases. These enzymes, in fact, typically increase the rates of chemical reactions by a factor of 10 to the 9 to 10 to the 20th power times relative to the reaction rate that would occur without that catalyst present. 10 to the 9, by the way, represents a billion time increase in rate. 10 to the 20 represents some number so large that I'm not even sure how I would go about describing the units of that. So the bottom line is that enzymes dramatically increase the rate of chemical reactions, almost like magic, but it's not magic, it's science. Enzymes are highly specific for particular reactants. In other words, enzymes recognize a particular type of chemical structure and increase the rate of reaction that that particular reactant will partake in a very specific chemical reaction. In other words, enzymes control reactions in a very specific manner. They aren't just generally going about catalyzing random reactions within the body. Instead, they serve very, very specific functions. And it's for that reason that the body has a huge plethora of different highly specialized enzymes to control the wide array of biochemical processes that we require for life at every second. Enzymes, in many cases, do not function alone, but instead require a type of structure called a cofactor in order to function. What's a cofactor? A cofactor is a molecule that binds to the enzyme and facilitates the function of the enzyme. And this is one reason why vitamins and minerals are essential to life. Many vitamins and minerals act as cofactors, such as the B vitamins, act as organic molecules that come in, bind to particular proteins, particular enzymes to be specific, and allow the enzyme to carry out its catalytic function. Without binding to the B vitamin, the enzyme is ineffective. Similarly, with a variety of metal ions, such as ones that are commonly marketed as supplements, such as zinc, magnesium, iron, nickel, and others, those metal ions bind to proteins at very specific locations of the three-dimensional structure of the protein and allow the enzyme to effectively carry out its catalytic function. So without these cofactors, enzymes cannot function in many cases. One way that we can recognize when we are looking at literature and analyzing what we're reading, we can often recognize enzymes based on the ASE suffix that we see in the name, such as here with our cute little illustration of an amylase enzyme. Amylase enzymes catalyze the breakdown or metabolism of a variety of different starches and carbohydrates, and that ASE tells us quickly that this is an enzyme. You might not know that that enzyme catalyzes the breakdown of carbohydrates, but by seeing the ASE, you recognize that this is a catalyst of some sort. Proteases, which we looked at in the previous video when we were looking at proteins in general and looking at their roles as catalysts, proteases are responsible for the breakdown of proteins, such as this chicken that this protease is holding here. Lipases break down fats or lipids, hence LIP there, breaking down this fatty piece of meat here, specifically breaking down the lipids from that, catalyzing decomposition of those molecules at rates that would absolutely not happen on a measurable scale if done without an enzyme. In the next video, we will look a bit under the hood at enzymes to understand how they work as catalysts for chemical reactions. For now, we need to become aware of some of the key terminology that we'll be using in that upcoming video. The enzyme, which I depict here, kind of Al Pac-Man style if you're in old school video games, binds via the active site to its so-called substrate. In this schematic, the active site of the enzyme has a three-dimensional shape that is complementary to the three-dimensional shape of the substrate. This is one reason why the topic of stereochemistry is critically important when thinking of enzyme function. If we change the three-dimensional shape of the substrate, for example, converting even one L chiral center to a D chiral center, then the substrate in most cases will no longer properly bind to the three-dimensional shape of the active site. So as we dig deeper into enzyme terminology, you should be aware that anytime we use the term substrate, we're referring to the molecule or molecules that bind to the active site for a reaction to ultimately be catalyzed by the enzyme. In other words, you can think of the substrate as the reactant, the enzyme as the catalyst for the chemical reaction, and the active site as the location where the action takes place or the chemical reaction is catalyzed, ultimately yielding through this the reaction product or products, at which point the enzyme returns to its original state so that it is ready to catalyze another chemical reaction. Because remember that enzymes are recyclable catalysts. After they've catalyzed one chemical reaction, they can be used over and over and over again. In the next video, we will talk more about how enzymes work, including understanding in more detail of how the substrate binds to the active site.